Today we're going to be discussing another modulatory system of motor control. This is the cerebellum, the squishy, funny little part in the back of the brain. So in this section of this course, as we're covering the different parts of the motor systems, we now have discussed the descending pathways from the motor cortex, voluntary control that modulates the activity of the brainstem and spinal cord in order to induce changes in muscle groups. We also talked about the reflexes that are involved within the spinal cord and the brainstem to induce immediate changes within muscles to react to stimuli in a very quick way. And we have now discussed the basal ganglia and the cortical basal ganglia thalamocortical loop, which is one of the major areas of the brain that allows for the learning of procedural memories, and it is a critical way that humans and other animals learn to use their muscles in order to do the things that they need to do to survive and thrive. We're going to add another part to this, which is the cerebellum. The cerebellum is an additional accessory area. It functions as a rapid corrective feedback loop that smooths and coordinates movements. So this is running alongside the basal ganglia, allowing us to perceive the world and create smooth motor output to get very, very accurate movements. So motor commands, they can issue muscles to induce contractions and relax in a coordinated way, but they have to be coordinated to ensure that the right amount of force is applied at precisely the right times, all depending upon feedback of proprioception, as well as overall external stimuli such as visual stimuli, letting us know that the kinds of corrections we need to make mid movement. That's what the cerebellum does. So what kind of information does the cerebellum receive? This is from a very old textbook showing that, and I've talked about how swinging a baseball bat is critically dependent upon the basal ganglia. Baseball bat is also a great way of seeing and observing that the cerebellum was going to play a critical role as well, because the input to the cerebellum is quite extraordinary. It's quite a range. It receives somatosensory input, proprioception. It receives visual input. There's auditory input. Vestibular input plays another critical role. So this is allowing us for sensing where our body is moving and where our head is moving. And as I've mentioned, the proprioceptive input. So this is letting us know where our body is in space. And the cerebellum uses all of this information in combination to coordinate movement. We don't think about our cerebellum all that much, and I would actually argue that it's unfortunately an understudied area of the brain, despite its critical importance. We do know that it actually plays a significant role in certain dysfunctional systems. So cerebellar dysfunction is associated with uncoordinated movement. We refer to this condition as ataxia, and a lot of ataxias are rooted in cerebellar dysfunction. So here's a brief overview of the cerebellum. Cerebellum is Latin for little brain. Bellum is is diminutive, and so it's little. And this, like cerebral cortex, the cerebrum is brain, and to call it cerebellum means little brain. Very cute. The cerebellum accounts for just 10% of the total brain volume, but as we talked about in the very first lecture of this course, it actually contains nearly 80% of all of its neurons. As I mentioned in the previous slide, it receives information from a huge range of brain regions, and including a ton of sensory information. And damage to the cerebellum, it impairs balance, as we saw in the previous slide. It also disrupts spatial and temporal coordination of movement and reduces motor learning, as we'll learn over the course of this lecture. So if we look at a cross-section of a slice of the cerebellum, you'll see that there are these layers. So the cerebellum has this laminar structure, and it seems like it's sort of broken up into different pieces. So within the layers, we see these very sort of pale areas, and then we have these dark purple areas. And since this is a Nissl or Kressel violet stained section, what we can see, those purple areas are cell bodies. And so that means there's a lot of cells within there. And if we zoom in, we see that there are these three basic layers. We have this very empty layer here, which we'll get to in a moment. And then we have the dark purple area here, which is loaded with tons and tons of little tiny neurons called granule cells. So this is the other kind of granule cell that I mentioned, like we see in the olfactory bulb. But in between this empty area and the dark purple area, we see these very, very large neurons, some of the biggest neurons in the brain. They're called Purkinje cells. We'll learn a little bit more about the details of the different layers in a bit, but I wanted to give you an overview of what the heck we're looking at when we see these different purple and sort of not purple areas. So it's also important for us to overgo the anatomy of the cerebellum. If we look at the exterior of the cerebellum, we see two sort of distinct features. In the middle is the vermis, and this is kind of a bump that runs down the midline. This regulates axial musculature through contributions to the ventromedial descending spinal pathways. Then we also have two cerebellar hemispheres. Here's one on the right, one on the left. And these control limb movements and contribute to lateral pathways. The exterior of the cerebellum is covered by a sheet of cortex. So this whole thing is the cerebellar cortex. It makes up the larger portion of it. It's got all these different layers in it. We have neurons that are very deep within the cerebellum. These are the deep cerebellar nuclei, and they relay cerebellar cortical output to other brainstem structures, in which we will talk about. The cerebellum, as we saw before, has this distinct structure, but there's, of course, like we're learning in a lot of brain areas, this actually can be broken up into other functional differences in different areas as well. So in the more lateral portions of the cerebellum, we refer to this as the cerebrocerebellum, and then we have the spinocerebellum. And then below, we have these areas here. These areas are known as the nodulus and the flocus, and they make up what's called the vestibular cerebellum. So we have three major subdivisions of the cerebellum that do more or less correlate with like being along the vermis and then along the hemispheres. So the cerebrocerebellum, the spinocerebellum, and the vestibulocerebellum is this portion here. If we unwrap it, you got the nodulus in the middle, the flocus on the outside, and that's the vestibulocerebellum. So the cerebellum is it's kind of its distinct lobe that sits on the back of the brain, but it obviously has to be connected to the brain, and it's connected through areas that are referred to as the peduncles. So we're looking at the cerebellum as if it's been cut off from the rest of the brain. You would have to cut it through the peduncles, and we have two major peduncles, and we have each, there are pairs of them and on either side of the cerebellum. We've got the middle cerebellar peduncle, through which the pontine nuclei connect up to the cerebellum. We also have the inferior cerebellar peduncle. And then this is the nodulus and the flocus, which makes up the vestibulocerebellum. So there's a number of routes that brain areas take to coordinate, to send input into the cerebellum. It receives inputs from frontal and parietal cortex via the pontine nuclei. So the cortex, a bunch of different areas within cortex, will synapse onto these deep areas called the pontine nuclei. And so these are within the pons, and they're on the ventral portion of the pons. Then these pontine nuclei actually cross the midline. So if you're talking about pontine nuclei on the left, they will desiccate and then connect to the right cerebellar cortex via the middle cerebellar peduncle. We also have another area of the brain called the inferior olive, which helps with coordinating a bunch of vestibular related information, as well as inputs from the spinal cord and vestibular nuclei that are ipsilateral in their nature. They go up through that green portion, which is the inferior cerebellar peduncle. So we have these two peduncles, which are basically axon tracts. They would be nerves if they were bigger and longer, but we refer to them as peduncles. And we have the pontine nuclei through the middle cerebellar peduncle, which is receiving mostly input from the frontal and parietal cortex. And then we also have these additional areas, the vestibular nuclei, the spinal cord, inferior olive, going through the inferior cerebellar peduncle to connect up to the cerebellar cortex. Now we're going to discuss the outputs of the cerebellum. So as I mentioned, we have these three cerebellar cortices: cerebrocerebellum, spinocerebellum, and vestibulocerebellum. The cerebral and spinal cerebellums, cerebella, they make up the majority of the cerebellum, and they connect to the deep cerebellar nuclei, which we saw in previous couple of slides ago, that are above the pontine nuclei. And so then this is the output from the cerebellum. And from there, we have different portions of the deep cerebellar nuclei. I know I've got them listed here, but it's not that critical that you know those. And they synapse onto premotor and motor cortex. They also will synapse onto some brainstem areas as well. This will help with the, so they're connecting all the way back up into the motor cortex. And this is how the cerebellum can contribute to motor planning. So for the ballistic movements that we need to do from time to time, where we're using maximum velocities and accelerations over a very short period of time, the cerebellum's instructions to the motor cortex, they're based entirely on predictions about outcome. So these kinds of movements are happening so fast that the cerebellum is simply making some calculations and then applying those to generate movement that should be coordinated as best as possible. So we know that these kinds of predictions that come out of the cerebellum, they're dependent upon past experience. So that means that they are learned. So this ability for the cerebellum to make these predictions about where a ballistic movement is going to end up, it's all dependent upon prior experience. And so the cerebellum is used in learning the precise coordination of what those movements are going to end up looking like. So therefore, the cerebellum represents an important site of motor learning. And it ascertains what is intended by a particular action versus what happened. And when this comparison fails to meet expectations, there are compensatory modifications that are made in certain cerebellar circuits to make rapid adjustments so that you end up with as close of an accurate movement as possible. Of course, one important thing that I should mention as well is that obviously there's the vestibulocerebellum, which synapses back onto these vestibular nuclei, which regulates lower motor neurons in the spinal cord and brainstem, and all of this is balancing over our balance, as well as regulating the vestibulo-ocular regulation, which I have a whole slide on that we're going to talk about briefly. So the cerebellum has at least 2 somatic maps of the body within it. Since it has this clear motor control, this is what that map looks like. I know it looks a bit alienish. And so within the spinocerebellum, we have a map that covers basically the entire body. And then we have on either side, on the lower ends of the cerebellum, on the more caudal ends, we have a map for one side of the body and a map for the other side of the body. So there's some processing that's happening within the cerebellar cortex. And so this is where we're going to start talking about these different neurons and the different layers within the cerebellar cortex. This is what I showed you before with these different layers, with the dark purple layer filled with granule cells. This is referred to as the granule cell layer. I mentioned these very large neurons. These are Purkinje cells. and they exist in the Purkinje cell layer. And then we have this area that's largely free of cells. There's some in there, but there are very few. This is referred to as the molecular layer. So these are the three different layers that make up the cerebellar cortex. And so what we're looking at here is basically one follis, so one of those little ridges, and we're zooming in, and then this would be the dark purple part here that's filled with granule cells. So they're only showing one granule cell here, but there would be tons and tons of granule cells. And then this is the Purkinje cells, these yellow guys. And then within the molecular layer, we're having connections for the dendrites of Purkinje cells. They have these huge elaborate dendrites that are actually quite beautiful. We have what are called parallel fibers that come out of the granule cells. And we'll explain that a little bit in a moment, but these granule cells, they send an axon that goes up, and then they extend a super long length along a follis. So along one of these ridges, they just go in a line like this, and there would be granule cells next to this that would have additional parallel fibers. That's why they're called parallel, because they run parallel to each other. And so you have these like long fibers that go along next to each other. You also have what are called climbing fibers that synapse onto the Purkinje cells. We also have mossy fibers, and those are the pontine nuclei that are synapsing onto the granule cells. In addition, we have basket cells. So these basket cells, these are cells that are within the molecular layer, and they modulate the activity of the Purkinje cells. We will be talking about this circuit over the next several slides. This is a little bit more of a schematic of exactly what I just talked about. Here I'm trying to emphasize these Purkinje cells. Purkinje cells have actually been really well characterized and studied for many, many years. Partly because they're very big, so they're relatively easy to see. They also have beautiful, like I said, beautiful dendrites. So this is a one Purkinje cell that's been filled. When you do a Nissl stain, all you can see is just the cell body, and so you don't see any of the dendrite. But if you can see the dendrites, you can see they're quite extraordinary. Really beautiful dendrites. Also, relatively to analyze. So for anyone who's interested in studying mechanisms that regulate how dendrites grow and how they branch, Purkinje cells are super great for studying those kinds of things, because they have these very elaborate and interesting dendrites that are really easy to see and really easy to trace. They also tend to be kind of flat. So depending on how you cut the cerebellum, you can get most of the dendrite, or if all of the dendrite, all in one layer of a slice. So you can get a really good representation of what the dendrite looks like, whereas usually a dendrite tends to be pretty extended within 3D space and not terribly flat. This is what a bunch of filled Purkinje cells look like. So these are Purkinje cells that have been genetically modified to express a fluorescent marker, and here they've colored it orange. And so you can see these big fat Purkinje cells, and these are the dendrites that are filling in the molecular layer. So a big portion of the molecular layer is filled with the dendrites of Purkinje cells. So this is what it looks like schematically. So walking through the different connections here. Those are all those granule cells making up nearly 80% of the brain. They're receiving the input from the mossy fibers. This is from the pontine nuclei. So the pontine nuclei are the inputs through the cerebellar peduncles, connecting to the granule cells, synapsing onto those granule cells. The granule cells have axons that go all the way up near to the surface of the cerebellum. And then that's when they split, and then they send out these parallel fibers. And these fibers are literally parallel. That's why they're called parallel fibers. And they run along a long axis, and they will make these incidental synapses onto a bunch of different Purkinje cells. And the Purkinje cells have these big elaborate fibers to receive input from parallel fibers from a bunch of different granule cells, as well as these climbing fibers that come from the inferior olive. So then the Purkinje cell has this integration of input that it's getting through the granule cells, as through those parallel fibers, as well as these climbing fibers. And then the Purkinje cell, it, like I said, is modulated by the activity of the basket cells, but it's the output of the cerebellar cortex. So the information comes in through the granule cells, as well as coming in through the climbing fibers. There's some calculations and modulations that are made here, with the basket cells playing an important role. And then all that information that's been calculated out is outputted through the Purkinje cells, and then they synapse onto the deep cerebellar nuclei neurons. So this is the cerebellar cortical loop. So let's look at that even more schematically. So we're breaking it down in further detail, and this is probably this portion plus this portion. This gives us some of like the general anatomical organization. This portion shows us the layout of these synapses and how they interact with each other. A little bit more detail about some of the connections here. So every Purkinje cell, a single Purkinje cell, it may receive input from as many as 200,000 parallel fibers. So you might have 200,000 different granule cells synapsing onto, sending its input to one Purkinje cell. Now, each of those parallel fibers is probably synapsing on multiple Purkinje cells, but a single Purkinje cell could be getting input from as many as 200,000 different granule cells. That's a huge amount of input. Now, each parallel fiber, it may contact as many as tens of thousands of Purkinje cells. So the vast calculations that are happening within the cerebellar cortex is huge. It's just enormous. It's really hard to fathom. There aren't too many parts of the brain that has this level of incredible connectivity across so many different cells, where a single cell might receive an input from 200,000 different parallel fibers from different granule cells, and then each of those granule cells may contact as many of tens of thousands of Purkinje cells. So the calculations that are happening in the cerebellar cortex is almost unfathomable. But the way it's connected here, so let's start with this more simplified version, because this will be a little bit easier to follow, and then I'll just go through it again through this portion here. So we actually have, so what I walked through before is called the cortical inhibitory loop. And that's with these projections from the deep cerebellar, or from the pontine nuclei. They are excitatory, hence we have a plus sign. And they are activating these granule cells. And these granule cells act as, they are excitatory as well. And then those will act onto the Purkinje cells. We know they're Purkinje cells because of the color here, right? We've got sort of peach and peach color. Purkinje cells are inhibitory. And so whatever modulation and activity is happening here from the inferior olive through the climbing fibers, as well as all the summed activity of the 200,000 or so parallel fibers synapsing onto one dendrite of Purkinje cells, all of that is leading to some sort of change that will lead to substantial inhibition. So this is a GABAergic synapse, synapsing onto the deep cerebellar nuclei. And then this is the output, which is going to lead to precise coordination of activity via the thalamus up to the motor cortex. So this deep cerebellar nuclei is then the output. And you can see this inhibitory synapse is very critical to this. Now there are pontine nuclei where you have the deep excitatory loop. Many of these pontine nuclei will also synapse onto other deep cerebellar nuclei cells. And then so there will be excitation coming through here, as well as inhibition. But the inhibition is primarily being driven by these Purkinje cells. So I don't want to get into the vast complexity of how this balance of excitation and inhibition leads to the coordination of motor activity, but just know that it does. But that inhibition and GABA plays a critical role in having this precise coordination, and it's through the Purkinje cell to deep cerebellar nuclei synapse in which that occurs. So we're going to talk a little bit about what happens when the cerebellum gets messed up. Cerebellar dysfunction. Cerebellar lesions, which can happen in a variety of ways, they lead to a number of motor dysfunctions. This can include, like I said, ataxia, which is uncoordinated and inaccurate movements. There's dysmetria, which is overshoot or undershoot of a target. Some of this can actually be easiest seen with a patient where that patient has to touch, say, here this clinician is using the tip of a pen, but this clinician could also put out just the tip of his finger and tell the patient, okay, touch this, and do it as quickly as possible. Going back, touching your nose, touching the pen, going back and touching your nose and touching the pen. Someone who is suffering from cerebellar lesions, you will see an overshoot or undershoot of a target. And so just really miscoordination. This is a nice way of doing this because it involves not just, say, a single muscle group, like a pair of extensor and flexor. It involves multiple because you have multiple joints involved. You got the joints within the hand, the wrist, the lower arm, the upper arm, the shoulder. So this is a coordinated muscle system unit. And so this is one quick way of seeing how well the cerebellum is actually working, because all of these muscle groups, these pairs, have to be working in coordination to get this precise movement, and it's all dependent upon also the visual input to get the targeting down exactly and directly. There's also something called nystagmus, which is rapid involuntary movements of the eyes. So this is another way of noticing if there's cerebellar problems. There are various tests that can be done to evaluate this as well. So when someone is showing appendicular ataxia, where there's cerebellar damage that leads to overshoot of a target. So this is a little bit of what we're seeing in the previous slide with the woman trying to touch the end of the pen. In normal conditions, if someone has a perfectly healthy cerebellum, they will observe a target, grabbing a glass. They'll be able to reach their hand out using, again, multiple muscle groups in order to do this, and squeeze the glass and grab it and move it. Someone who's having appendicular ataxia will have very uncoordinated movements when they need to do something like this. So this person may be just sitting there perfectly normally, but when they're called upon to do some kind of task, like, say, grab a glass, they may wobble and not be quite on target. They may overshoot and then eventually grab the glass. So this is one major feature that we can see with cerebellar ataxia. There are more symptoms in cerebellar dysfunction that can be found at this YouTube link. So if you're watching this via the PowerPoint, you can just click on this link so you can watch the video there. I will also have a link to the description, and you can also look for it up here. You can just click right here as I am moving. There should be a link up at the top someplace. It should be right here. Yeah. Other problems that can happen with the cerebellum. Well, it turns out the cerebellum is a major target of alcohol. Those who suffer from alcoholism and have abused alcohol, one of the first major features that you can see in the brain is degeneration of the cerebellum. So this is what it looks like in a patient who has had degeneration of their cerebellum. The other thing that we can see is that, because we're talking about coordination of movements, and if you think about the connection of alcohol and movement, is that it can be used as a way of assessing whether someone is impaired and when they shouldn't be driving. So most of these tests that one has to do when they're being tested for whether they are legally drunk or not by the police, like you get stopped, walking along a straight line, taking your hands, putting them out by extended out by to your sides, and then touching your nose with a single finger, and they'll tell you the left and the right, and you have to do it. They're looking to see you mistargeting, like you're showing some sort of appendicular ataxia. Now, clearly, you're not like having any significant damage, but if you have taken on too much alcohol into your system, one of the first places that you're going to see some of this dysregulation is going to be cerebellar dysregulation. And there's no way to really compensate for that. And so you will be stumbling and not be able to walk along the line, and you won't be able to target your nose. And that's because alcohol acts on GABA systems. And we talked about how we have the Purkinje cells as a major inhibitory output of the deep inhibitory, cerebellar inhibitory loop. And if you have substantial inhibition happening via activation of those GABA receptors because of alcohol, then you're going to have an imbalance of that excitation and inhibition, and you're going to get uncoordinated motor output. And so because you have so many of these neurons and the Purkinje cells are so sensitive to alcohol, having all this additional GABA changes because of the alcohol, you're going to have enhanced GABA tone within the cerebellum, leading to uncoordinated movement. So, yep, don't drink and drive, kids. There are some patients out there that are particularly sensitive to alcohol. And this will show up as ataxia. So this is some data from a study, 1998. An individual, a 46-year-old man, who suffered from cerebellar ataxia-like symptoms every time he drinks, ever since he was in his teens. So this person cannot drink at all because he ends up with these ataxia-like symptoms. And so for individuals that are like this, a small amount of alcohol will induce gait and speech difficulties. So this is beyond just like simple slurring and stumbling. It looks a lot like ataxia, and it doesn't take a whole lot to do that. With this individual, they gave him 5 grams of alcohol. And just for reference, a 12-ounce 5% beer has 14 grams of alcohol. So just a 12-ounce 5% beer, which is pretty typical nowadays, has 3 times the amount of alcohol than what this individual was given. And within 20 minutes, the subject's gaze evoked nystagmus, slurred speech, and leg tremor, very much symptoms that are indicative of cerebellar ataxia. He couldn't walk without help after just having 5 grams of alcohol. And a kinematic test showed that the patient could no longer perform a simple wrist flexion task. So in this case, what this person had to do is they had to extend their wrist a certain amount. And so they would have to extend the wrist approximately 15 degrees. So you're moving your wrist, and it's a very simple task. You put your arm out, and all you have to do is bend your wrist 15 degrees to hit a target. And this is the movements that this person was able to make with alcohol. This is showing how long 100 milliseconds is. And so for this person to do it with complete accuracy, going from 0 to 15 degrees, took around 300 milliseconds, which is what it would take for just about anyone to do this. You can see there's a little bit of overshoot and small corrections, but it's all, hardly anything noticeable that would be unusual. When this person was given 5 grams of alcohol, you can see is that there's this huge overshoot, which is very typical of cerebellar ataxia. So they go too far, and then they go down below, and then above and below. And so this individual, it would take them, probably about a second or so in order to get their wrist in the correct position, and with a huge overshoot. So not getting just to 15 degrees or say 16 degrees, going all the way up to sometimes 25 or 30 degrees. So almost double. With just a tiny little bit amount of alcohol. So whatever was going on with this individual, clearly something genetic, leading to this person to be particularly sensitive to the GABAergic effects of alcohol, and so likely affecting their Purkinje cells so that they could no longer do these tasks. So that's exactly what's going on here, right? We've got this loop with this inhibition from the Purkinje cells, and that if you have a GABAergic input, it's going to be like you have from the Purkinje cells. You're going to have dysregulation of that inhibitory output, which will affect the output of the deep excitatory loop, and this will be far too inhibited, leading to this ataxic-like symptom where you have these major overshoots. The cerebellum also can control and contribute to saccadic eye movements. And this happens via at this point, our favorite midbrain nucleus called the superior colliculus. So we're going to learn about the output of that. So some of these deep cerebellar nuclei, they project to, we have projections to vestibular nuclei, reticular formation areas to control the lower motor neurons, leading to precise coordination of the head and neck. But there's also inputs to the superior colliculus as well. And so we're going to learn a little bit about some of those connections from the deep cerebellar nuclei. Again, these are the outputs of the cerebellum. So they're located here. They're deep within the cerebellum, right? We've got the cerebellar cortex way out on the outside with this branching like pattern looking to it with the different folia. And then you have these deep cerebellar nuclei, and some of them, there are outputs that connect to the superior colliculus. So we're going to talk about one experiment in which this was done in monkeys, where there was a weakening that was induced within the eye system. So the right eye was normal and unaffected, but what they did to the left eye is that they surgically weakened how well the lateral rectus pulls onto the eye. And they did that by just slicing the tendon a little bit so that it couldn't pull quite as effectively on the eye. So if you do this in a monkey and you have the right eye open and you have a patch on the left eye, but you still are able to track the left eye, if this muscle has been weakened, and this is right after the surgery, there's going to be a signal sent to the medial rectus on the right eye, right, and the lateral rectus on the left eye, right? We learned about this in visuomotor control. So they're both receiving a signal to contract. If the right eye is open, that signal is going to be told to say, hey, move this much. You're going to move, something like 6 or 8 degrees, and the eye is going to move 8 degrees to the left, and then it's going to be right on target. And this is a saccade, and it happens super quick, as we learned. But what you see in the left eye when it's patched is that it can't meet its target because the signal that is being sent is now insufficient. The amount of contraction that's happening in this muscle is insufficient to pull the eye completely over. So it doesn't reach its target. It misses. But because there's a patch on, the monkey doesn't know, doesn't even see. It thinks it's doing just fine because it can't see. Okay? But now let's see what happens. When you move the patch to the right, so now we've covered up the right, the monkey is told, okay, here's a target, you got to move. And the right eye hits its target, right? Because it's patched, and there's a signal that's sent that says, okay, this is where we need to move. From the superior colliculus, giving a motor output signal to say, move your eyes this much to the left. But it's the left eye that's now running the show here. And because that tendon is weak, the signal is insufficient. And then the eye needs to catch up. So it does these catch-up saccades where it's trying to get to the target, and eventually it does it enough times where now it's really pulling on that lateral rectus, and it's now at this position. But now, because it's impossible for this monkey to not do this, it's sending, every time it needs to move the left eye, it's sending a similar signal to the right eye, and none of it's being corrected, so that the right eye is now going way too far past where it should. But there's no visual input, so it doesn't know that it's messed up. When you patch the right eye for this long, eventually the monkey will learn, and it'll be providing a more intense signal with this weakened lateral rectus. So it's going to give a much bigger signal saying, hey, move this much. And the left eye will eventually hit the target. It doesn't need to do it in a stepwise fashion because after 5 days, it's learned, okay, well, things are pretty messed up. It's pretty weak. So I'm going to have to provide this kind of signal to this eye. And then all of a sudden, I'll be able to hit my target. But because the right eye is patched, Now we've got a huge displacement, and the eye on the right is now ending up in the completely wrong place. And it's now, instead of being like right at 8 degrees, it's now 25 degrees off. So there's some interesting things happen, though, when you lesion the cerebellum with this process. When you are undergoing these changes, these learned changes, if an individual has a cerebellum that's lesioned for the parts that are controlling the eye here, it never actually stabilizes. So on the left, this is a monkey with an intact cerebellum, and this is the normal eye, the eye that's on the right that's been patched, and you can see it's able to hit its target perfectly. Boom. So its presumed target. It's patched, so it can't see, but you can see it's perfectly flat. The weak eye is also able to hit its target as it needs as well. But one thing that you can see is that the weak eye in the monkey that has the lesion cerebellum is that in the weak eye it's still able to hit that target because it has the same visual feedback. But for the other eye, the signal, this corrective signal that has been learned over time, this lesion in the cerebellum has led to a very imprecise targeting. And it's led to the target to overshoot and not actually fully stabilize. And so the cerebellum, this is an illustration that the cerebellum can be used or is used to provide for new changes that have happened within the muscular system in order to learn and accommodate to those changes. So this is a form of kind of a neuroplasticity that under normal conditions, the eye will end up in adjusting and creating a new target, but doing it very precisely. But if the cerebellum is messed up, it cannot do it precisely. It's going to be overshooting and never stable, and instead will be wobbling. And so that's where the loss of the cerebellum is most obvious, is that it ends up leading to a messed up targeting system that is not completely stable. So this is a demonstration that the cerebellum is critical for fine motor control and adaptation. So this is what, and really is where the cerebellum plays a major role. So the lesions to the cerebellum, they block adaptation to the vestibular ocular reflex plasticity system. I am not going to expect you to memorize this circuit, but it includes some of the things that we have already learned. We have the ocular motor nucleus, the abducens nucleus. The abducens nucleus connecting to the lateral rectus, ocular motor nucleus connecting to the medial rectus. And this is showing that we have also, there's a vestibular portion that regulates this, but all of this also being driven through the cerebellum. So these are the Purkinje cells and the parallel fibers and the granule cells, and this input coming via to the granule cells to the parallel fibers, calculation through the Purkinje cells, leading to changes downstream that then allow for accommodation of the abducens and ocular motor nucleus in order to have this change. And the vestibular regulation of the vestibular ocular reflex is dependent upon having an intact cerebellum in order to do that kind of behavior. Again, I've mentioned it a few times, but we don't have to have the circuit memorized. But I just wanted to kind of show this because for some of you that may want to work out exactly how that is occurring, well, it looks like the cerebellum would actually play a critical role in some of these other additional areas that lead to control of the abducens and ocular motor nuclei to accommodate for this change. And if you eliminate this part that changes the precise timing to the abducens and ocular motor nuclei, then obviously things are not going to work very well. We're going to walk through another experiment that shows that the cerebellum plays an important role in accommodation to changes within visual input and its impact on eye and head movement. So we've talked about the vestibular ocular reflex. That is the reflex that keeps images maintained on the retina. And so if a monkey, or yourself, you're just kind of a big monkey yourself, but a monkey in this case, is asked to maintain its gaze on a target. So, and you can do this too. Just look at a dot, and then you start turning your head to the right and to the left, but you're keeping your eyes fixed on that target. Your eyes are going to counter-rotate in the opposite direction of the direction your head is moving. So if you turn your head to the right, your eyes counter-rotate to the left. And they have to move approximately the same amount in degree. Right? That's exactly how it has to work. In order to keep the eye's gaze perfectly in line on the target. This is called the vestibular ocular reflex. Now let's say you put minifying glasses onto a monkey. What are minifying glasses? Well, some of you may actually wear minifying glasses, but in this case, they're really, really bad. But minifying glasses, this is the kind of glasses that nearsighted people need to use. And this is an individual with some pretty severe minifying glasses. And you can tell that they're minifying glasses because when you look at the individual's eyes inside these glasses, they actually look really tiny. So hence they're minifying. And so what happens often with these experimental kinds of minifying glasses is that the eyes don't have to move as much because the visual field is now being compressed and then and put onto the retina in a much smaller part. But we have already our vestibular ocular reflex. We've learned it over the years and years and years that we've been alive on how to do this. And when this monkey starts to turn his head, the eyes, unfortunately, are moving too much when they're being asked to stare at a particular place. And it's because the motor signal that's being sent to the eyes is too much for the image that's being transposed onto the eye because of the crazy glasses that this monkey is wearing. So the eyes move too far in relation to the image movement onto the retina when the head moves. So when the head moves, the eyes are going to end up going to the wrong side. And eventually the monkey will correct. But the initial movement is too much because the signal coming from the cerebellum is too much. But after several hours of doing this task, you can reset the gain on the vestibular ocular reflex. And so now the eyes, they actually move a smaller movement. See, you can see this arrow is smaller so that it can maintain the position so that it's facing directly forward. And so that compensates for the head movement. So the head can move as much as it needs to, but the eyes don't have to move as much because of these crazy glasses. Now, cerebellum seems to play a role in that. How would you even know that? Well, we know that if you have lesions to the cerebellum, monkeys cannot do this. So very similar to the kind of experiment that we saw before, where the eyes are unable to have this reset in the new position when they're being patched and when one muscle has been weakened, where the other eye, the healthy eye, cannot reset, and it has this wavering signal that happens. The same happens for this vestibular ocular reflex gain. The reset cannot happen, and so these monkeys are never able to end up with the smaller eye movements when they're wearing these glasses, and instead they continue to overshoot. Again, illustrating the importance of the cerebellum in maintaining proper motor signals in order to get coordinated movements. So lesions to the cerebellum, they end up blocking the adaptation for the vestibular ocular reflex plasticity. So if you block this, when you destroy this, you're going to interfere with the kind of gain that the gain changes with this. You know, this is seeing clearly the cerebellum is seeing the mismatch here because it has the visual input coming through the inferior olive, as well as and so that's referred to as retinal slip. And when you lesion this, now there's no calculation happening within the cerebellar cortex. And so none of that is happening, and you can't have that output signal allowing for the consolidation of the signal for the brainstem to provide the proper signals that are needed for the abducens, the ocular motor nerve, to allow for the compensatory movements of the eye. It just doesn't happen. So when the cerebellum is lesioned, the output to the brain areas that control the eyes do not receive the corrective signals that are needed to make the precise movement, and the eye fails to make the correct patterns. Once again, you don't have to have this circuit memorized, but I'm putting it on here so that you can appreciate the connections of the cerebellum. We learned about this circuit, and we did learn about, or at least you'd seen, the vestibular ocular circuit, and you are familiar with these areas. So I thought you might appreciate the overall connections here and how this might be influenced. So to wrap up, here's some key questions about the cerebellum. What is the role of cerebellum in controlling movement? What kind of information is relayed to the cerebellum? What is the basic anatomy of the cerebellum? What are the connections from and to the rest of the brain? So via the peduncles, what comes up through the peduncles? What are the major structures of the cerebellum? What are the major neurons that make up the cerebellum? How are they connected to each other? How does information flow through this circuit? This is all one point here, and it's all about a detailed circuit. Hint, hint. What is cerebellar ataxia? What is the role of the cerebellum in other conditions, such as alcohol use and abuse and regulating eye movement? And we talked about, basically half of what I talked about today is what happens when the cerebellum goes wrong. And so it's going to be important for you to know some of those outputs and the clinical connections to that. All right, so that's it. We'll see you next time.